Welcome everyone to our May installment of the Knowledge Portal Network webinar and workshop. I hope everyone's well and healthy. Sorry about the dog. Sorry about that. It wouldn't be home working if you didn't hear a dog. So, Iris, I want to get started with just some little announcements. I'm share my screen. And there we go. So today, I'm just going to give a couple announcements at the beginning about upcoming developments and what's to come over the summer. And then I'll turn over to Maria, who's going to talk you through a lot of the new feature and sort of functionality changes we've made in the portal in the recent release. Last time we talked about the human genetics calculator that's now on the portal. And we talked a lot about the data and the different types of things, but we didn't talk about the enhancements to the reading page and new features. So we're going to talk about that today. So as always, Hayden, can you stop playing with her? Resources and webinars. Our webinars are every other month. Our next one's coming in July, and that will be actually about the lung disease portal, which will be fantastic. Michael Cho will come talk about that. And our following one will be in September. As always, in between these, we have resource tabs, which include our videos, documentation, tutorials. And a new thing that we're implementing is an opportunity for our contributors to participate in videos. And this is really, you want to hear from the source. So the person who's actually doing the method or the data or contributing to the feature on the portal, we're actually going to have them do a little video to kind of demo it from their own perspective from the research. And Peter Donbass is going to do the first one for the human genetics calculator. We're going to have more of those coming on. So you'll get another series of videos that will sort of give you another lens into how we represent the results and the methods on the portal. Another forum we're thinking about is something, these webinars are really about us talking to you and telling you about features and data and things like that. But really, we want to sort of flip that on its head as we sort of move to the next installation of AMP CMD as we grow in the partnerships and in the portal to a mechanism for user engagement that's slightly different. And then we're thinking about this running about bi-monthly in between the webinars. And we're thinking this will be basically a forum for you as users to give us feedback, weigh in on new features and investments. We're going to show you things half-baked, show you things where they're messy and somewhat new. And we're also going to use this as an opportunity for you to tell us what you think we like. So we're going to come with sort of like, this is what we're thinking, and get your opinions and your feedback. This is really going to be an open forum and really meant to be a working session, not necessarily us just presenting to you. And so the first one we thought would be really good for that would be GEM, which is where we encapsulate LocusZoom in the portal, and really an area for active development for us, which is to allow you to browse a genetic association in the context of credible set, annotations, enrichments, and so on and so forth. This is something that we plan to build out in the next couple of years, but there's many directions we can go with it. And so we want to basically use our investments appropriately and carefully. We want to talk to you about what you think are the next best steps. So this next one, we're scheduling it roughly for June. We'll send out an announcement about it so you can all get access to it if you're interested. But we really want someone to be willing to talk on the call rather than listen to us. And our developers from who build the LocusZoom module itself will be on, along with our front-end engineers, to talk about what are the features, what are the things we're thinking about, and then hear from you. So look forward to that in June, and that'll be in between our webinars in terms of timing. A couple things on the horizon. I'm just going to touch upon these lightly so you can see what's to come over the summer. And then Maria is going to talk more specifically about what's on the portal now. I want to pay attention to this space. This is a place where we actually are putting all of our new features for the most part, unless they're classically embedded into the portal. Maria's going to talk about this in depth today, but again, this is this research space. This is a place where novel research, novel data sets, methods, and tools can be placed in the portal for people to give us feedback and iteration before they're incorporated in the fuller sort of workflow of the portal. And this enables feedback in real time, also as a place for unpublished work to be placed. You're gonna hear a lot about that today from Maria, but a couple things that are coming online for that may be of interest to this group specifically is that we've released a beta complications association browser. And that's been really, nice to have, but it hasn't had a lot of data in it. And we're excited that later this month, we're going to have some large-scale complications US from Ronnie Salem coming for 18 complications in the context of type 2 diabetes that you can view and browse in this framework for May. Again, we mentioned this before, lung disease knowledge portal is now live and online. That's going to be an area of active development for us. What's very cool about that is that in May, we hope to integrate that with a resource that our colleague Kyle Galton, who also builds the Diabetes Epigenome Atlas, contributed to for Lung Epigenome Atlas, and that will be fully incorporated. So annotations that are relevant to lung disease will be incorporated in the knowledge portal and represented in May too. So that's quite exciting. Some more teasers. I mentioned the Focus Forum, which is a chance for you to give us feedback, unvarnished feedback, we hope. Also, we haven't talked about this portal in a long time, but the ALS portal that we have on the portal is now going to be updated to our new framework that many are used to. But in addition, we're going to be bringing to that portal as a sort of a space to start developing a neurodegenerative disease portal. We're going to bring in Parkinson's disease, Alzheimer's disease, and other associated psychiatric and neurodegenerative diseases into that framework in the coming months. So look for that. That should be exciting. In terms of our common metabolic disease knowledge portal, look for a new wave of data coming up, including kidney disease, liver disease, and many more phenotypes in that space. Also, we're continuing to build out, beyond just the genetic data, beyond just the genomic annotations and representation of those, really the culmination of what we're learning about effector genes for type 2 diabetes and its complications. And for that, we'll be adding evidence sources and new trait results for effector genes on the portal over the coming months and years. One specific one I wanted to call out, we mentioned this before, is a curated effector gene list from the coronary artery disease group that is going to be released with publication. And that should be very valuable, I think, to add to the other ones that are up there for type 2 diabetes and whatnot. Another thing we're exploring is, ways to represent other genetic models, non-additive models and pleiotropy. We don't have those on the portal currently. We have several collaborators, including Ronnie Salem and Joseph Mekador, who are interested in doing that and who developed papers and approaches for that. So we're hoping to integrate that in the portal the coming months and weeks. And finally, new mechanisms for access and ways to work with the platform. And what I mean by that is not just, you can get our data programmatically through APIs, but things like if you have your own data, how would you bring it to the portal? How would you interact with our system? How would we allow you to run methods and get it just on your data? So more programmatic and more user-driven ways for someone to customize their portal experience. And this is a long haul. This is something we're thinking of the next couple of years. These are just some teasers to give you an idea of where we're headed. We welcome your feedback in between these user forums, directly emailing us, call me up, do whatever works. We're happy to give your feedback because I think that really drives the ability for our portal to grow. So with that, I'm going to turn it over to Maria after I thank this wonderful team that does all the work and does all the things that you see here. And Maria is going to walk you through some of the exciting new things on the region page.
All right, let me share my screen. Okay. Yeah, as Noelle said, I'm going to focus on the region page today. We've recently made some changes to the way that we process data, and they are affecting some displays in the region page. So I want to go through that in some detail to make sure you understand it. So I'll be using the Common Metabolic Diseases Knowledge Portal as my example, but this applies to all of the portals, all of the disease-specific portals that are embedded within the CMDKP, and also the independent portals, lung disease and musculoskeletal knowledge portals. That this is the navigation and the content are analogous for all of these portals. So let's go to a region page. When you enter a gene name here, and I'm just going to click on the example to save time, you get to a region page that covers the coding sequence of the gene plus 50 kilobases on either side. And you can quickly expand that in 50 kilobase increments if you want. And if you want to do something more complicated or even navigate to a completely different region, you can enter your coordinates or parameters in here to do that. So the top of the page is meant to give you an overview of what's going on with genetic associations in this region. And as many of you know, if you've already used the portals or heard this type of webinar before, most of the associations that you see in the knowledge portals are meta-analyzed. So for any given phenotype, we may have many, many data sets. Like for type 2 diabetes, we have dozens of data sets. And rather than presenting them all to you independently, we meta-analyze the results. And we use the METAL algorithm developed at the University of Michigan, and it accounts for sample overlap between data sets. So this gives a really nice consensus view of the associations for a particular phenotype across the genome. So we've been doing that for a while in the portal, but new with this latest release, we are also performing LD clumping. So the idea behind this is that for any given associated locus, there's probably one causal variant, maybe more than one, but usually one. And it's the variant that makes the DNA change that actually impacts the phenotype in some way. But of course, there are many other variants close to it, and they're genetically linked to it. And these will also have really significant genetic association scores, even though they're basically just a long. Yeah. Sorry, question? Okay.
Yeah, Jerry raised his hand, Maria, actually.
Oh.
Just let him, hold on, how do I do that? There it is. Hi, Jerry, what would you like to ask?
Well, first of all, how do I know which gene this is?
Okay.
I'm looking at this page. I don't see any gene.
Well, this shows the genes that are in this region. It's not just one gene. It's a region that includes all of.
So you picked a region or you picked a gene?
Well, I started with a gene on our homepage.
Which gene did you start with?
I just clicked on our example gene, PCSK9.
Okay, I'm sorry. You went. I missed that. Okay, that's the next first thing. Okay, then the next thing when you get to, I may have some questions about your LD clumping, but keep going.
Yeah.
Okay. Good point, Jerry. You can basically pick a gene, a region, or variant and get to the same framework. What it does is it anchors to whatever you put in and goes 50 kb.
And the statement about not overlapping, that doesn't come from metal. Doesn't that come from the bottom P line project?
Accounting for sample overlap that's how we do the bottom line meta-analysis.
Yeah. Okay. It's not metal.
Yeah, but Jerry, but it's actually metal. Metal is what implements it. So there's an option in metal to account for overlap. So metal is like the tool that's used as part of the bottom line workflow.
Okay, great.
Okay. Okay, LD clumping. So yes, the point of LD clumping is to simplify the view. So that instead of seeing many variants just for the most strongly associated phenotype, you're seeing clumps of variants that are each associated with one phenotype. And I wanted to just quickly show you how we do LD clumping in the portal. And this, with the caveat that I'm not an engineer, so but luckily Jason's here to answer any questions about this part. But we do LD clumping using the Plink software, and this is just kind of a really high level basic outline of how it's done. So for each phenotype, Plink looks at common SNPs, and those are those that are found in the 1,000 genomes reference panel. And it looks for those with a p-value for association with that phenotype of less than a variable we'll call p1. And so P1 is explained down here. It's initially genome-wide significance, 5 × 10⁻⁸. But if there are not at least 50 SNPs that have that significance, then the value gets multiplied by 10 in increments of 10 until at least 50 SNPs are found. And it only goes down to there's a bottom value beyond which it doesn't go of 1 × 10⁻². So these associated SNPs are found, and then Plink looks at their linkage disequilibrium, or LD scores, relative to the other variants within 250 kb. And so LD is expressed as a value called R². And so the Plink clumps all the variants whose R² is greater than equal to 0.2 with those SNPs that were first found. And then it assigns a lead variant to each clump. And then as a final step, we look at rare variants, and those are those that are not found in the 1,000 genomes panel. And if there are rare variants that have a p-value under that p1 threshold, whatever it was for this particular phenotype, then we assign those as single variant clumps.
What do you do about conditional analyses? So within a given linkage disequilibrium clump, one can have more than one variant that is significant.
Yeah, so those are all clumped together. there. So there's no conditional analysis performed, but it's like kind of a poor man's version of that, right? So you
No,
take
it's a poor man's version based on LD clumping. I get it. If there's different LD groups, so to speak, or haplotype blocks, whatever term you want to use,
right.
then this will find the lead variant for each. Yeah, I'm not saying what you should or shouldn't do, or not acknowledging the constant evolution of the knowledge portable, which is from a person who doesn't work on it but only observes it, quite remarkable to me. So, I mean, I'm just clarifying. So at the moment, you get a lead variant for each clump, but you're not doing conditional analyses within the clumps.
No.
Okay.
No. Exactly. So it's almost like the appropriate way to do this would be to do like an approximate conditional analysis in the region, right? And then you would get kind of a better measure of the independent signals. But this is kind of assuming that you take, all the variants in a clump are the same signal. So yeah, so there's no condition analysis of any kind.
Okay, let me ask another related question very much on this that didn't occur to me before. These traits are from multiple ethnic groups.
Yes.
This is a summary meta-analysis, so it includes simply every ethnic group together.
Are you going to address that, Maria, or should I right now?
No, why don't you do it?
Okay, so yes, that's actually, we should add, I don't know if this is in the documentation on the site, but that's really the non-standard thing that we're doing here. This is, on this slide, is just the standard plain clumping procedure. That's all we're doing. What we do is we run that for each ancestry separately because it requires a linkage to disequilibrium estimates, which of course, as you say, are population specific. We then get a set of clumps for each ancestry, and then we essentially merge clumps across ancestries such that if a variant is shared in any, at least one variant, right? So you get a set of clumps in each ancestry, and then you merge them across ancestries such that if any variant is shared by the clumps, they get merged together. So it's essentially like a meta-analysis, if you will, of the clumps, right? But we're not doing the clumping on the trans-ethnic results. We're running the clumping.
You're doing it on the individual. Now that all relates in the what is defined as the African origin populations will have smaller
right.
areas than and even other ethnic groups. It's not always smaller. It's often it's different in one ethnic group versus another. How are you accounting for that in this?
So we're not accounting for that, right? I mean, it's just I would say we're being overly aggressive in terms of merging clumps. So if you have a very small clump in Africans, right, and then there's two SNPs and one SNP overlaps a much larger clump in the Europeans, those two clumps will be merged together.
Okay, so, just so we know what's going on. Is that explicit in the document if people want to find that out rather
than that's what I don't know.
Yeah, that is not. I have to add that to the documentation.
Right. I mean, it's one thing for me to ask you the questions, but of course the advantage of the knowledge portal is a person can come in and learn it by themselves.
Right.
Okay.
Exactly.
So this is not yet a tool. The knowledge portal is at this stage not a great tool then for comparing ethnicities to learn things.
Not at this moment, no. But that
is these are not negatives. These are just
that's fair.
questions.
Yeah, it's fair. That's a fair assessment. I think that's something that
what's the state of the art? As I say, I'm constantly amazed at what it can do and what you add. I'm just trying to clarify where it's at any given time.
Yeah, indeed.
Mm-hmm. That is actually though something that this is the first method that we've implemented that has ancestry specific results. We have another that we have implemented and run, but not made on the portal. COJO, the joint conditional analysis, which also requires ancestry level. We're going to be implementing LD score regression soon. So it's becoming apparent that in the near future, the portal will need to start exposing ancestry level results. And if anybody has feedback on that, we don't want to overwhelm people with information, but that's certainly something we'll be adding in the near future.
People can always have links. You can say, you can be very explicit. This is a summary result, whether you call it transethnic or just a summary result. If indeed you want the ethnic specific, you go to this subsequent page. You don't put it on the first page.
Right.
Use it as a sub. I agree with you that there is a nice utility. I mean, I'm looking at this figure, for example, very nice utility on the figure right on top. That is great for the region. When you say the clumping, But I noticed here, these are not, so in other words, the the lead variant for LDL cholesterol is not the best lead variant for total cholesterol.
Right.
Interesting. Interesting in that regard, based on these summary meta-analyses, bottom line p-values, etc. Okay, I mean, that's some interesting questions there, but that's not to answer this. And what would be, well, I guess it's obvious. If something is all the same lead variant, it's simply the same number for the various traits, like coronary artery disease is the same lead variant as IDL, for a whole bunch of them in a row. So, okay, I got it.
All right, Maria.
Okay, so just to explain this display a little bit more. So the graphic here shows the lead variants for clumps where there's genome-wide significance or better. And it shows the top 10, but if there are more, you can click on a button here and show them all. And for some regions there, you'll have to do some horizontal scrolling on the screen because there are many, many genome-wide significant hits. We have an alternative visualization here, just a different way to look graphically at the associations. And this groups the associations by phenotype group, just a broad category. And then you can click on each one and it'll expand to show the associations for individual phenotypes.
Oh, that's good. And so the color is just because orange is lipids and green is CAD. Is that
That's
it?
right. That each group is color coded. Yep. And there's a key here to the colors. So then in the table, it shows basically the same thing. Each row is a clump and represents a clump. And so you see the lead variant with its values. The green here is just kind of a bar graph that represents the differences between the p-values here. So you can see at a glance, how they compare to each other. And then if you want to see the individual variants in the clump, you can click on this button and it will give you a table that has all the variants, and some of these have many pages. You can page through them and see what's in the clump. Okay, so that's this part of the page. And now if we scroll further down the region page, everything below this point is phenotype specific. And I want to explain that because it's a little bit confusing. So when you go to a region page, there's a phenotype automatically set for the page. You can call it the primary phenotype. That is the phenotype for which there's the highest association in this region. So for this region, it's LDL cholesterol. And that sets these default displays from this point on the page down. Let me just show you what they are. First of all, there's a table of the top variants. Now, these are not clumps. These are just individual variants for this phenotype. And it's nicely filterable. I'll show you that in a little bit. And then also below it, in our genomic region minor module, there's a locus-zoom plot shown by default for that same phenotype. I'm sure you're all familiar with locus zoom, but a new feature that we've added to it from our Michigan colleagues is this track up here that shows whether if for the variants that are present in the locus zoom plot, it shows whether those variants are represented in the GWAS catalog and for which phenotypes, and you can link to GWAS catalog or DBSNP. So this is a nice quick way for a variant of interest to seeing whether there are other associations to it. So that is the default display for the primary phenotype of the page. And if you want to change the primary phenotype, there are a couple ways you can do that. Either right here, where it's showing you the primary phenotype, you can exit out, and then you can choose a new primary phenotype here. And then now the table and the locus zoom plot will be set for that primary phenotype. Alternatively, if we scroll up to the top of the page again, everywhere there's the name of a phenotype, if you click on it, you get options, and set below to this phenotype. I think we have to come up with a better way to express this, I think, but that sets the primary phenotype. So I've just set it back to LDL cholesterol, and now below this point, everything we're seeing pertains to LDL cholesterol. Now, I know that many of you like to compare multiple phenotypes, and especially to stack up locus zoom plots for various phenotypes across a region and see what you can see. So there is a way to do that. After you have a primary phenotype, you can add a different phenotype, and you can just start typing in here and then select the choices that correspond to it. So we've added total cholesterol here. And the way this works is that the set of variants that is shown in this table is the top variance set for LDL cholesterol for the primary phenotype. And then for each of these variants, we've looked to see what is the association for total cholesterol. And that's what you're seeing in this column. So it keeps the same set of variants, but looks at their associations with total cholesterol. And down here, we've also added a locus zoom plot. And so you can look at these two. You can, if you mouse over, you see that you get these nice crosshairs where you can line up things between plots and see if they correspond to each other or not. Okay, and one other feature I wanted to show was that we have filters here. So if you click in consequence, you can see all of the predicted consequences that there exist for this set of variants. And you can just pick one that's interesting. And it filters down the list. And then you can also filter even further by closest gene. So here we're looking for variants that cause a stop in this gene, and there are only 3 variants. And the neat thing is when you filter this table, it also filters the locus zoom plots. So we only filtered down to 3 variants, so we can barely see them here. But I think you get the idea. You can hone in on a set of variants that are really interesting to you, and then see them visually as well. So, okay, I think that is what I wanted to say about genetic associations. But now, of course, there's a whole nother dimension, epigenomic data, and that is available in GEM. So to do that, let me, I'm going to go back to I'm going to go to a different gene. Let's go to a different region, CBC 1, 2, 3. So here I'm navigating to that region. I'll expand it, just make it a little bit bigger. So again, here we see these are the genes that are in this region. And we've got the clumps and the significant associations summarized up here. And this page is now set to type 2 diabetes as the primary phenotype. So this GEM module is meant to allow you to explore not only genetic associations, but also credible sets and epigenomic annotations, which come to us from our sister resource, the Diabetes Epigenome Atlas, or DGA, which is led by Kyle Galton at UCSD. So let me show you how some of this works. So first of all, let's yes.
Quick question from Jerry. I'm going to unmute him.
All right.
You should be able to talk, Jerry.
Yeah, on the previous one, you went from, for example, LDL of 10 to the minus 300 and a total cholesterol of 10 to the minus 18. The question is, for anybody who would be interested, that's the sum of all evidence. But of course, the sum of all evidence is somewhat proportional to the sample size from which those are derived. In other words, if the 10 to the minus 300 came from 1,000,000 and the 10 minus 18 came from 1,000, the 10 minus 18 is actually more powerful than the 10 minus 300. How would anybody know? Is there a way that somebody can find that out?
Yeah. So for each phenotype, we have a phenotype page, and you can link to it like this, or you can just search for it from the home page. So like here's our phenotype page for LDL. And if you scroll down this page, it shows the data sets that went into the meta-analysis. And so there are multi-pages here, but for each of those, you can see the sample size. So does that answer your question?
Yeah, that's actually terrific. Does it sum that? Well, maybe I can see why it might not, because you're using different kinds.
There's sample overlap, so it does.
Yeah, then there's sample overlap. Okay, well, that's exactly my question. So that anybody who wanted to could find out on what the data is based on. That's
great, but like we don't have the effective sample size and things like that yet. And
no, I understand that.
We're
but
trying to get
even a sense. I mean, that's of course ideal, but there are many things that are ideal. You're not going to accomplish them all at once. Just that somebody could get a feeling for it. You know, if they went in for total cholesterol and it was the same sample sizes,
yes,
then they would know the relative power of the effect. So that's good. That answers my question completely.
One other just quick comment on that. One thing that we've played with is to allow users to not show p-values, but to show percentiles. within the trait, which would be another way of sorting if you wanted to look at the, you know, to
What do you
basically
mean, what do you mean
normalize.
percentiles in the trait?
Yeah. So like the strongest, like what you're saying, the minus 70 p-value, if that's like the #1 variant, right? That might be more interesting than a minus 305 if that's like the 1000th variant, right? And it's just everything scaled up. So essentially correcting for sample size, right? P-values corrected for sample size is a view that we're considering adding to toggle on if people would like to sort things that way.
But you said something about percentiles.
Yeah, well, so you just convert the p-value, you know, you order all of the variants.
Oh, you're ordering the p-value. So this is the #1,
Yeah.
#1 or number 10 or whatever
Right.
for the trait, with the percentile being the LD clumped p. You're going to use the LD clumped as
That's
your
why we
variant?
haven't done it yet, because it's not a standard thing, and,
Right.
and we'd be happy to take thoughts on it,
Yeah.
that people have. I don't want to.
Yeah, you need to do some kind of, ideally, you would have your lead variant and your conditional variants, because
Right.
it's not unimportant to say that if the first 5 variants were all in PCSK9 and you only got to LDL receptor with number 6, or the first lead variant is in PCSK9, the second variant is an LDL receptor, and then you go back to PCSK9 in terms of relative effect. I mean, you could do it at the current level with LD. You certainly don't want to do it with all SNPs because that's kind of useless, right? 500 SNPs in a region, who cares? So you don't want to do it with all SNPs. I mean, I like your idea. I like your idea a lot. The question is where to implement it. And I guess imperfectly, if you're talking about gene-centric, so to speak, doing it by one lead SNP per LD clump, and some genes will have more than one LD clump, so that gets partly to the conditional, so to speak. So that, and as we said before, I continuously acknowledge the portal does certain things, and other things are in evolution, or one can't, as human beings, do everything at once. So I like your percentile idea, and you could use I would use your current version of your LD clump versions as the percentiles, meaning this is the highest SNP, this is the second best SNP, etc. But with the idea eventually that once you do put in conditionals, that they get ordered in there among the percentiles.
Okay. All right. Maria, take it away.
Very clever idea. I like the percentile idea a lot, actually. I like that.
Definitely an idea.
I just didn't understand. I thought it was like you were saying percent contribution or something like that.
Yeah.
That's why I didn't understand it until you.
All right.
Okay, so to get back to GEM, just want to show you a little bit more about how to navigate GEM. And first, credible sets. So you can add credible sets to this display. And we have 3 kinds of credible sets. We have credible sets that we've computed just from our current associations, and those are available for every phenotype in the portals. Then we also have manually curated sets from the Diamante European GWAS study from Anuba Mahajan and colleagues. And then we also have a set of credible sets for 40 different traits that were generated in Hilary Finucane's lab at the Broad. So you can choose any of those, and then you'll see them on this display. And for this one from Diamante, there's just one variant with very high posterior probability, and it seems to line up with the index variant here. So that looks kind of significant. So that's a way to explore credible sets. And now I'm going to remove this panel just to have a little more room here on my laptop screen. And now let's go to epigenomic annotations. So as I said, these come from DGA, and we get them at the portal, and then we process them using the Gregor algorithm currently, which looks for enrichment of genetic associations for phenotype within tissue-specific epigenomic annotations. So for example, if associations for diabetes are enriched within a potential regulatory region in pancreatic islet tissue, that could be, that's good to know, potentially very significant. So that's the idea. And as Jason said, we're going to be processing these using LD score regression in the future. We're working on that. So we've recently made some changes that make it a lot easier to navigate these data. So in the tissue menu, formerly we used to list every very specific tissue and cell type on its own, and there was a list of, I don't know, hundreds of very specific types, but Kyle at DGA had curated that list to group it into broad organ groups, organ and tissue groups, so that this is much more easy to navigate now. So we'll choose pancreas. And now we're seeing annotations for pancreas. And okay, it's a pretty colored bar, but the way to get some meaning out of it is to click this button, split tracks. And now what you see is a track for the annotations in pancreatic tissues of all of these different types of epigenomic data that we have. Most of these are chromatin states, which are predicted regulatory regions. And there's also accessible chromatin from techniques like ATAC-seq, looking for open chromatin. And we are adding more data types to this list too. So you can see these regions, and you can use the crosshair to line them up with variants of interest. And another useful thing you can do is that you can filter these annotations by a p-value or by fold. So because Gregor looks at enrichment of associations in these regions, it assigns a p-value and a fold enrichment. And so let's just filter by twofold. And you can see this really kind of clears up the display and gives us only the most significant or more significant enrichments. And now if we line it up, we can see that in pancreas, this variant is actually located in a region that's predicted to be an active enhancer, both type 1 and type 2. So that's pretty interesting. There's another way to look at epigenomic annotations. If you don't know which tissue you're interested in, but you know what type of data you want to look at, you can use this menu. And now annotations are grouped by the data type. So let's look at accessible chromatin. And again, we can split the tracks to see things. And so the filtering is still applied to this. We can remove it. Remove it, you see there's a whole lot there, so it's probably better to put it back. It'll simplify our display. And now if we scroll down, if we line up that index variant, we can see that it is in accessible chromatin in the eye. Well, it's not really clear what that might mean, but it's also accessible in the liver, which is a very relevant tissue for type 2 diabetes. So this variant, I didn't find this on my own. This is a variant that has been studied by Kyle and others, and it actually changes the transcription factor binding site that affects transcription in pancreas and I think in liver too. So it's that's the reason for these associations. But this is example shows you the kinds of things that you can actually find out using this display, and we definitely want to improve this, add lots more data types and make the navigation easier. And yeah, as Noel said earlier, we're very, very interested in your feedback on the best ways to do that. All right, so those are the main changes to the region page. And I thought I would quickly go over some of the new tools in the KP lab space before we end. So since we've been talking about LD clumping, I want to start out with a new tool called the Signal Sifter. Those of you who used to use the portals probably remember a tool that we had called Variant Finder. And this tool let you look for individual variants that were associated with one or more phenotypes, and you've got to set the criteria for their association parameters. Do we have another question coming up?
Just
Yes,
in the tissue, for example, you said the tissues will come like pancreas, but of course, depending on your interest, pancreas is exocrine pancreas, endocrine pancreas, it's islets, it's subset of islets, beta cells, alpha cells. Is there a way to disaggregate that?
Not at the moment, but that's definitely on our list. Yes, we realize that's an important thing to be able to do.
That's my question.
Okay. So for the Signal Sifter, it acts a little bit different from the Variant Finder in that it doesn't look at individual variants, it looks at clumps. So for example, you can find a clump that's associated with a variant of interest, with a phenotype of interest. And then the Signal Sifter will look to see if there are variants within that clump that are associated with other phenotypes of interest. So let's go through and show you how it works. So we can start with BMI. So when you first select a phenotype, you get a Manhattan plot showing the clumps, and you get a table. Now again, each row is a clump, and then you see association parameters for the lead variant. And then you can add phenotypes. So let's add type 2 diabetes. All right, so when you do that, now the Signal Sifter has looked within this clump to see if any variants in there, or to find the top variant associated with type 2 diabetes. And so here, for example, here it is. It's not the same as the lead SNP for BMI, but it's in that same clump. And you can repeat this. You can add multiple phenotypes to find, clumps that contain variants associated with multiple phenotypes. And you can also filter this down. So for example, maybe we want to find variants that have a positive effect on BMI, but a negative effect on T2D. And then, so we've narrowed down, and now that's what we're seeing. So here's a clump where there's variants with those parameters exist. So that's the idea behind the Signal Sifter. And you can
One point I'd just like to clarify in case anyone, I know we got a question about effect allele. All the effect sizes have been aligned to the lead SNP in the clump. So even though, as Maria is showing, the top signal here, well, actually it's the same variant, but the second signal here are two different variants. The effect alleles have all been aligned to that lead variant in the BMI clump. So you can do queries like increased BMI, decreased T2D, and those queries will work even if the variants are not the identical variant.
Good point.
All right. Any more questions on the Signal Sifter? Okay, well, I'll touch quickly on some of these other KP Labs tools.
Jerry has a question.
Okay.
It would be fascinating to have a quick look at the 9P region with CAD and type 2 diabetes. I'm curious what the since we've been thinking a lot about that, what the portal shows when it does that.
All right, do we have time for that in terms of your?
But
Yeah,
if you don't, we can save it for another time or an individual discussion. But
Yeah, no,
I think that would be a particular. It's just because we've been thinking about a lot of how many are in the regions. Is there a type 2D region? Is there a CAD and type 2D, et cetera, et cetera, et cetera. So a bunch of questions related to that region. But if you can save it for another time if you want.
We can just do it quickly. You were looking for CAD, and what was the other phenotype?
Type 2 diabetes.
Okay.
In the 9P21 region.
I think you're going to have to go to the.
Oh,
there
we go.
you go. It is the top.
There we go.
Top one.
Yeah. Wow. Okay.
You can click on that. You want to click on the. Remember, these are p-values. I think by default these are genome-wide significant.
Yes.
No, that's not true actually.
No,
I'm actually surprised.
No, they're not all genome-wide significant.
You're right. I think it's because
Oh,
any
no.
cardiovascular disease is probably not a very good phenotype.
How about coronary artery disease?
Oh, yeah, you're right.
Yeah, that's what you want. Right?
This is a phenotype
CAD.
that doesn't have many.
Yeah.
Yeah.
Good point. That's gonna have a bigger number, the coronary artery. Yeah.
Yes.
That's better.
There we go. Okay.
So help me. Yeah. Okay. So
There's a couple rows here, a couple clumps on 9. There's this row, and then there's this row.
yeah. That's. Well, I see, because it gives it by anyone. So if you just wanted to focus on 9, you have to pay attention to the numbers below and on top. But you see, oh, can you do a zoom on 9?
Well, this will go to the region.
Yeah, that will bring you to the region page. So that you might have to do some of this on the region page where you can see multiple phenotypes at a given time.
Okay. Well, let's.
Jose, you raised your hand. What's up?
I just wanted to make sure, there's a lot of people attending this. So, there's somebody, Jerry, if you have a personal interest in a specific area of the.
That's what I said. We could do this individually another time.
Sure.
I'm
All right.
okay.
Let's
Let's
go,
do that.
let's go back to KP Labs. Wanted to just quickly mention that, the Human Genetic Evidence Calculator, which was a subject of our last webinar, is now online for you to play with here. I'm not going to go through it because we had a whole webinar about it, and the recording is available from our website if you missed it. But so that's there for you. And then another KP Labs thing that we've just updated is genetic loci clustering. And this is work from Miriam Udler's group. And so we had this 2018 data. We've had this on the site for a while. And this is her original study where she took 94 loci that were associated with T2D and then clustered them by their associations with other traits. And you could view all of the supporting evidence here. I'm not going to go through this interface right now, but with the update, this is work from Claire Kim and her group and Miriam. They've done a much bigger clustering analysis now. So they've looked at more than 300 T2D loci and also loci for coronary artery disease and other things. And so that's all available here again with all the supporting evidence. And there's a very detailed method that they provide so you can really understand exactly what they did. And accompanying that is an epigenomic enrichment that was done by Josh Chu and Kyle Galton's lab. So they've looked at these clusters, and so here are the clusters on the left side, and then you can see their enrichment in all these various tissue and cell types. So that's pretty cool, and there's research details for that as well. Let's see, also in KP Labs that's new, we have some preliminary data from an exome analysis of over 86,000 exomes, and this has gene level association scores for type 2 diabetes, and you can select masks to filter out the variants. I'm not going to go into this in detail right now either, but that's also available. And I think that is basically what I wanted to talk about. Those are the new things in KP Labs. Yeah, I think that would be that. I think I'll stop there and take questions.
As Maria mentioned in KP Labs, there's also the Human Genetics Evidence Calculator, what we went over last time, is there and available. I think you mentioned that too, Maria. And then complications data is going to be updating at the end of this month. And then, one of the things we mentioned, which is an ongoing area of development for us, is effector genes. And we didn't talk about that particularly today, but under the tools lab, if you're interested in that, as a reminder, you have the 2 prediction methods on the site and also the curated one. And those are all subsumed in the effector summary that's right below there that brings all of them together into one table that you can sort and filter and review. Just as a reminder to you all, this is probably one of the areas of active development where we want to get to for many phenotypes within the portal and really enrich the portal, no pun intended, for effector genes for diabetes and its complications. And then at the end of this month, as I mentioned, we'll have a brand new release with lots of new data and other updates to these features and some new things in GEM, which will also, beg the forum that we're going to have in June to talk about what the best directions for that are, because we have lots of people actively interested in what types of tracks we want to visualize there, how you would use that as a means to step through to start prioritizing variants based on a region that you're interested in. So that's really something that we're hoping to get more feedback on. Again, I think, Jerry, you taught us today that we should be having more of these forums where we can hear from you as the users. So that's really useful for us so that we can start to plan more targeted conversations with people who have really good expertise in that area. So that was really helpful to think about it from that perspective. Other questions or comments from folks? Right. Next month July will be Michael Cho and our colleagues from the lung genetics world who are working with us to build that portal. Actually, we had some nice conversation with them, and they're going to come speak about their perspective, why they want to build the portal, what it's useful for them, and then we'll walk through the data and the results that we have in that portal as well at the next webinar, which is in 2 months. And also, some interesting, exciting developments. AMP T2D is moving on to AMP CMD, and so you'll hear from us in July and June about new partnerships and new directions with that partnership, which is very exciting to us. And we're very thankful for that continued growth in that project space. So this time was about, features and tools. We also want to keep the science rolling. So again, you'll hear a little bit about lung genetics next time, and you'll hear a little bit about the portal, and then we'll be continuing to evolve. The forum will be specifically about user feedback. Also, Maria and I have toyed you with some ideas about setting surveys. I know people don't like to fill out surveys, but it's a way for us to learn what you're using the portal for. I think that, one of the things that occurred to me and Jason as we're starting to demo this, is we need to make sure other camps, if you will, in the community are getting access to this tool and knowing what we can and can't do with it. And so if you all, as users and people who, I often see at webinars, which I appreciate, have ideas of places where we could be, tapping into a new community of people that we could demo the portal from, that would be really great to hear as well. So with that, any other last comments or questions? Thanks for the active discussion. And with that, we'll see many of you in June for the forum, and I'll put that on everyone's calendar so you can join if you want to. And then we'll see you in July for the official webinar. Thank you, Maria. It was great.
Thank you. Thanks, everyone.
Right.
Bye-bye.
Bye, folks. Take care.